Assalamualaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is enzymes. So in this tutorial video we will discuss about the nature of enzymes and different parts of an enzyme, different terms of an enzyme such as apoenzyme, holoenzyme and some other informations. So let's start with what is an enzyme. Students, an enzyme is a biological catalyst, a living catalyst, which can accelerate and speed up a specific chemical reaction. The speed of that particular chemical reaction is increased when the utilizing enzymes lower the activation energy of that particular process. So these enzymes can increase the speed of a chemical reaction but remain unchanged, unaltered during the chemical process. In simple words, we can also say that an enzyme is a living substance, a living structure that is almost present in all the cells of all the organisms, which can increase the rate of a chemical reaction. For example, during glycolysis and Krebs cycle, a large number of enzymes are utilized, such as hexokinase, phosphoglucoisomerase, phosphofructokinase, and various other enzymes. All of these enzymes accelerate these specific chemical reactions, these specific biological processes like glycolysis and Krebs cycle. And almost all the procedures that are occurring inside a cell can be speed up with the help of enzymes. So we can say that the enzymes only increase the rate of chemical reaction. However, they are not utilized in a chemical reaction, and also their presence does not affect the nature or chemical properties of end products. So this is the answer of that question, that what is an enzyme. Next, we will see the structure of an enzyme. For understanding the structure of an enzyme, students, we will say that an enzyme is made up of proteins. Suppose this is a simple diagram that is representing an enzyme. So this is our enzyme. Inside this enzyme, you will view the amino acids, a large number of amino acids which combine to each other to synthesize the structure of an enzyme. Many students ask that from which substance the enzyme is synthesized, as an enzyme is a very active and efficient material inside the cell. So it is necessary to know about the enzyme structure, its function and role of an enzyme, whether it is made up of carbohydrates, lipids or proteins, because it is even made up of by the combination of anything. Answer is that proteins, meaning amino acids. So enzymes are protein in nature, meaning that the enzymes are synthesized from proteins. Which components the proteins are synthesized from? As I have already prepared two videos of translation process, during which I have clearly described that the proteins are formed from polypeptide chain, and a polypeptide chain is synthesized by the attachment of amino acids. So when hundreds of amino acids are attached to each other, then these prepare a polypeptide chain. This whole mechanism is described at our YouTube channel. If you want to watch the topic, then you can, from which you can clearly understand that the whole mechanism of polypeptide chain or protein synthesis. As these round circles are showing amino acids, many amino acids mean a polypeptide chain which represent a protein. Almost all the enzymes are made up of proteins except one enzyme, that is ribozyme, which is an exceptional case that is made up of nucleic acid. Because of this, we shall say that all the enzymes except ribozyme are prepared from proteins. So most of the enzymes are protein, but some are nucleic acid like ribozyme. Dear students, this chemical nature of enzymes is divided into two parts.
The first part include those enzymes which are prepared made up of only proteins, namely amino acids. The second type consists of those enzymes which possess not only amino acids but also consists of some other chemical groups. Let's see, this is the chemical nature of enzymes, which comprises of two types. Consider first type. First type consists of only those enzymes which are synthesized only from amino acids, that are known as simple enzymes, that are occurred only 10% in nature. Certain examples of simple enzymes include trypsin, chymotrypsin. Now consider the second type. As you have known that amino acids combine with some other chemical groups, because two different chemical groups, like the first is amino acid and the second is some other chemical groups, joined together are preparing it. So these are called conjugate enzymes, that are about 90% in nature, that we will study in our next upcoming video in detail. Dear students, all of the enzymes that are present in our bodies, out of which 90% are conjugate enzymes, whereas only 10% enzymes are of simple enzymes in nature that occur in our bodies. Dear students, these simple enzymes are only of protein nature, made up of only amino acids. So these enzymes consists of only polypeptide chain. As this enzyme consists of only amino acids, so it is a simple enzyme. On the other hand, we can understand a conjugate enzyme with the help of this diagram, as conjugate enzyme is synthesized with the help of a protein part and non-protein part. By their combination, imagine that this is a protein part and this colored part is representing the protein part, and when we combine this large protein part with this non-protein part that is in smaller quantity, so by the combo of these two, protein and non-protein part, we shall achieve this conjugate enzyme, prepare this conjugate enzyme. I hope now you are clearly understand the terms of simple and conjugate enzymes. Next, you will have to see that in a conjugate enzyme, 95% of amino acids will combine to prepare structural unit of a conjugate enzyme. On the other hand, functional unit is prepared by the combination of only 5% of amino acids. As you have known that what is the main function of an enzyme? Yes, to perform a catalytic activity, to catalyze, to speed up a chemical reaction. In this catalytic activity, all the amino acids of an enzyme do not take part, but only a short number of amino acids participate in the catalytic activity. Let's suppose if a conjugate enzyme consists of a number of 129 amino acids, as mostly enzymes consists of 100 or more than hundreds of amino acids, and 100 or above than hundreds of amino acids combine to prepare a conjugate enzyme. In this number of amino acids, only the 5 units of amino acids will be the functional units. Rest of the 124 units of the amino acids will combine to prepare the structural unit. Is this clear? Next, see this, that is the functional unit. This 5% functional unit is only able to perform the catalytic activity of an enzyme, participate in the function of an enzyme. Students, the basic function of an enzyme is to participate in a catalytic activity to speed up a chemical reaction. So it attaches to a substrate and then either splits it or synthesizes it, forms it. Students, whole of the enzyme does not take part in the catalytic activity, in the function of an enzyme. So which place is in the enzyme at which enzyme performs its function and plays its role, because all of the enzyme structure does not play its role in function. Students, an enzyme consists of only a specific site at which this enzyme perform its function by attaching to a substance. So what is this substance? The name of this substance is the substrate. Substrate means anything at which enzyme will act. So what is the function? What is the job of an enzyme? To split or form the things. If an enzyme will act on a protein, carbohydrates, then it will be a substrate for this particular enzyme. But if an enzyme acts upon a fatty acid molecule, then fatty acid will be a substrate for this enzyme. That's why anything upon which enzyme acts will be a substrate for this particular enzyme. You can write as a substance upon which an enzyme will act is called a substrate. That is the definition of substrate. Here a question emerges in our minds that which place is on the enzyme where a substrate will be attached. The name of this place, this site is active site. Active means function performing and site means place. So this active site is the function performing place of an enzyme. Students, why whole of the enzyme is not active? The answer is that chemical reactions cannot happen upon the whole enzyme, upon the throughout the enzyme. So complete enzyme has only one place at which catabolism is occurring, metabolism is occurring, which is active site. So this place is the active site of an enzyme. At this active site, some charged amino acids are present. About these charged amino acids, it is said that these will form our active site. The active site is itself of two types. The first one is named as binding site and the second site is named as catalytic site. So now divide this active site into two sides. Students, now we see what is the job of binding site. As its name is representing, it attaches substrate. Not only it binds a substrate, but it also recognizes that this substrate is mine or not. So to identify or to bind a substrate, these are the two functions of the binding site, which finally forms an enzyme substrate complex. This reaction also activates the catalytic site. Next, we see in addition to it, what does do the catalytic site? Students, catalytic site either forms a substrate into product or split it into smaller fragments. So we can say that catalytic site catalyzes a chemical reaction or the reaction is completed at this catalytic site. So this activated catalytic site change the substrate into products. So now we have known that active site consists of two sites. At this binding sites, enzyme identify and binds the substrate to its place. And at the catalytic sites, enzyme performs its job, that is to catalyze a chemical reaction. So these are the two sides of active site. In one book, I saw that 2 to 12 amino acids are present in the catalytic site, but in another book, it was written that 2 to 12 amino acids lie in the active site. And also note that these amino acids are charged amino acids because only charged amino acids have the ability to bind with a substrate. Next, we will see how an enzyme substrate complex is formed. Suppose this is our enzyme has denoted by E and this is our substrate has denoted by S, where the substrate will be attached at the active site. So by the combination of enzyme and substrate, it will become a combo of enzyme and substrate, that is called enzyme substrate complex. Students, imagine after the formation of this complex, what will do the enzyme? For example, enzymes prepare and split the things. So for example, here enzyme splits the substrate. Consider it has broken down and the enzyme itself is free as the same it was before. Now these broken pieces of a substrate are now called as products. I hope now you are completely understand the substrate and product terms. Substrate is that substance on which an enzyme will act, whereas the splitted or synthesized form of a substrate is known as product. Students, substrate is also named as reactants. and those new materials which are formed after the enzyme's catalytic activity are called products, and after the catalytic activity the substrate is converted into products. Students, it is the overall mechanism of an enzyme. Dear students, you must also know that this is the main body of an enzyme which consists of globular protein. Dear students, these rounded circles are showing globular protein, which has a large number of amino acids. These amino acids are attached to each other with the help of peptide bonds. These lines are showing peptide bonds in between these amino acids. So, enzyme's body represents a tertiary protein configuration. Therefore, we can see that a polypeptide chain of this enzyme folds upon itself to form a globular protein, and this polypeptide chain of tertiary structure is strongly interacted by disulfide bridge or hydrophobic interactions. So we can say that this globular structure of enzyme is very important, is very unique for its suitable job, its proper functioning, that is to catalyze a chemical reaction. If this globular structure of an enzyme is lost, then enzyme will destroy. So a globular part is a very important component of an enzyme. This globular part of an enzyme possesses at least one surface region which consists of a pocket-like hollow space. This pocket-like hollow space consists of only a small portion of the enzyme surface, which is termed as enzyme's active site. Dear students, active site of every enzyme possesses a specific shape. Therefore, a substrate attaches into it in a very specific way, in a very unique way. Weak chemical forces like hydrogen bonds are created which hold this substrate into its active site. Students, this is the first part of our enzymes video. In the second part of this enzymes video, we will discuss about complete enzyme, cofactors, coenzymes, prosthetic group, holoenzyme, and apoenzyme. In addition to these terms, we will also conversation about types of enzymes like intracellular and extracellular enzymes. In that video, we will also study about allosteric site of an enzyme. At the end, I want to encourage you that take your education seriously. That is very important to you. Many thanks for watching this your video.